This is Club EvMed. I think most of you or a lot of you have been here before. I'll just quickly go through some of the background details while more people are tuning in. Club EvMed is organized by a bunch of different evolutionary medicine centers. I'm Charlie Nunn. I'm at Duke University. I run TRISEM. That's the Triangle Center for Evolutionary Medicine. And we're one of many organizers of Club EvMed. And in addition to those centers, we also work with the International Society for Evolution Medicine and Public Health, and I'm pasting all of this into the chat right now. And I want to just call out three things very quickly. Please consider publishing your work in the Society's journal, Evolution Medicine and Public Health. We also have two special issues, one of them involving cancer, exploring EVO applications in cancer. And very importantly, I want to call your attention to our annual meeting. It's coming up in July, at the end of July, July 28th to the 31st in northern Germany in Kiel. It's going to be a great location. The plenary speakers are all selected. Everything's really lining up nicely. I hope you'll consider joining us for this meeting this summer. And with that, I'll say again, thank you for joining us. And I'm going to turn things over to my colleague, Jason Somarelli, who's actually the organizer of one of those special issues, the one on EVO applications and cancer, and my colleague here at Duke. Go ahead, Jason. He's going to
Thank-
introduce the speakers.
Thanks, Charlie. Yeah, submit your papers. Yeah, I'm like very excited to introduce this stellar team of Doctors Graham, Lockley, and Whiting. Dr. Trevor Graham is director of the Center for Evolution and Cancer at the Institute of Cancer Research in the UK, where he leads a team that's seeking to understand the evolutionary dynamics of cancer development and also translating this knowledge to improve clinical management of disease. Dr. Whiting's a postdoctoral fellow at the Institute of Cancer Research, and his work combines both theoretical and experimental systems to define the evolutionary dynamics of cancer therapy resistance. And then we're going to hear maybe a more clinically focused topic with Dr. Lockley. Dr. Lockley is a professor and group leader at Bart's Cancer Institute at the Queen Mary University of London. And her work focuses on understanding how cancer cells and their tumor microenvironments co-evolve during therapy. So that's something I want to pick your brain about later. We've been thinking quite a bit about that, but you're not here to hear from me. This is an all-star cast, and I'll turn it over. I know they have a great program for us. So thanks to all of you, and please feel free to take it away.
Thanks, Jason, and hello, everyone. It's a real pleasure to have this opportunity to talk to you all. So we want to talk to you about thinking about evolution in cancer treatment and how bringing in evolution into our thinking might help make things better. So I've got a really brief introduction about why we need to think about evolution, and then I'm going to hand over to Freddie here, who's going to talk about thinking about putting evolutionary thinking into the drug discovery process itself. And then finally to Michelle, who's going to talk about using evolutionary theory to optimize the way we do clinical trials and use the drugs we already have better. So quite a diverse lineup for you. So let me give you a little bit of introduction. I'm going to share some slides now. There we go. Hopefully that's all good. All right. So opportunities to use evolution to improve cancer treatment. So first of all, a few disclosures. The only one that's a little bit relevant for what we're going to talk about today is the last one on the patent list there. So let's think about cancer. So my background is in mathematics, and I like to think about things as simply as they can possibly be. So in my head, this is what a tumor looks like. It's made up of cells. They're the circles there. And essentially, for any given drug, we can think of there being two kinds of cells, ones that are killed by that drug. They're the blue ones in my diagram, and ones that are not killed by the drug. They're the resistant cells. They're the orange ones in my diagram. So what happens is, of course, if a tumor has resistant cells at the start, and we think it almost certainly will do, because there's huge numbers of cells in tumors and they're very genetically unstable often, so there's lots of genetic diversity there, we'll apply our drug and it will kill the sensitive cells. And then we can keep treating, of course, but all that's left is cells that don't respond to the treatment, the resistant ones, and of course this tumor will slowly grow back. So this is the evolution of drug resistance. And this is why often our treatments work for a while and then stop working. And so this is the problem we need to try and tackle. I want to show you a really specific example of this. This is a clinical trial published a few years ago in lung cancer, where a new wonder drug had been made. This drug targets the KRAS oncogene. As you likely know, KRAS is thought of as the key driver of lung cancer. The vast majority of lung cancers have a mutation in KRAS, and it's thought to be the critical driver of the disease. KRAS was also considered an undruggable target, and so some truly remarkable chemistry has been done in order to make drugs to hit this target, and we're finally able to drug the ultimate driver. So there really isn't a better target to have a cancer drug against. But yet, sadly, when we look at the benefit of using the drug in this large phase 3 randomized trial, there's only a 2-month benefit for progression-free survival, and there's absolutely no statistical benefit about overall survival. So despite hitting the ultimate target with incredible technology, this wonder new drug, there's really marginal benefit here. And we suspect that's because of the evolution of resistance. And so if we go back a bit in the development of this drug, this is the original publication of the first phase 2 trial of the same drug, again in lung cancer. Back to back in the New England Journal was the publication of the phase 2 on the left, and then in the same issue, the examination of resistance mechanisms that evolved to this drug. So it's really not a surprise to anyone. People knew when they were publishing this that resistance was going to evolve to this drug. So there we go. So this is the situation that we have, and we want to tell you about how we might be able to use evolution to deal with it. So again, here's a tumor on the left, a mixture of sensitive and resistant cells. I think there are 2 key areas where we have an opportunity to intervene. The first one is in the molecular causes of resistance itself. So what causes a blue cell to become orange in my diagram and be resistant to drug? And Freddie's going to talk about how we might be able to do better thinking about evolution in these molecular causes of resistance. But then there's a second opportunity, and that's about the evolution of the tumor itself. And this is what Michelle's going to speak to. You know, glibly, it doesn't matter if there's one resistant cell in the body. One cell's not a problem. But what is a problem is when that first cell becomes very many cells and there's a burden of disease, which we can't treat anymore. And so we think there's unexploited opportunities to dig into those evolutionary dynamics and hopefully improve outcomes. And so Michelle will speak about that. So that's me done, and I'm going to hand over to Freddie. He's going to talk a bit about evolutionary thinking in drug discovery.
Yeah, so thanks, Trevor, and hello, everyone. I'll be talking today at a kind of general level about our thoughts on how we can incorporate some of these evolutionary ideas into drug discovery. And in the interest of time, I won't go into the technical details. So if you're interested in those, you can, of course, well, you can read the papers, or hopefully there'll be an opportunity you can email me with specific questions as well. So I'll start off with this kind of highly idealized, simplified version of what we picture the drug development process as. So as I'm sure you all know, it's a highly risky and expensive process where we start off with some promising cancer drug target on the left there. You know, there's a huge effort to try and get to the compound, which then goes through these preclinical studies. And this is a kind of iterative cyclical process to improve the potency and safety profile of that compound. And ultimately, we end up in clinical trials. And that's for the very few drugs that make it that far. And as Trevor just highlighted, we see this all too often, this common trajectory whereby we might see an initial response in patients, but inevitably we see drug resistance evolve. So we started thinking about, is there something we can do earlier on in the drug discovery process to try and mitigate resistance evolution? And we think there's more that can be done preclinically. So thinking, you know, coming up with more clever ways to think about drug resistance during the preclinical process. And I'll give an outline of our version of what that would look like today. And so in the long term, this is kind of how we picture the process changing. So instead of waiting, you know, for years and billions of dollars to just observe resistance evolve in the clinic, we could actually start with information on resistance to the drugs we think we're going to take forwards and incorporate that information into the development process itself. And ultimately, of course, the goal of that is to minimize resistance in patients. And so this is a schematic trying to highlight what we think one of the problems is at the moment, and that's that a lot of preclinical assays focus on the short-term response to a drug, but But of course, there's nothing saying that the potency with which you can kill cells on a 3-day window has anything to do with how robust that response is over time. And when we talk about how robust the response to a treatment is, we just mean what's the propensity for resistance when we continuously treat with that drug. So, you know, a lot of work's been done here on the short-term response to those drugs, and focusing a lot on the differences we see here. And we think a lot less attention has been given to differences down this end of the schematic. So looking at the time the time until resistance emerges to that given drug. So, you know, hopefully that's suggested the rationale for developing preclinical techniques that allow us to quantify drug resistance. So, and these are some of the features that we think such an assay would need to have. So we think the ability to actually quantify resistance evolution, so a kind of quantitative, not just a qualitative approach, is important. Obviously, being able to then subsequently identify the resistance mechanisms themselves has repercussions for biomarkers or new drug targets. And then this is a more recent focus of ours, but we think the ability to identify cross-resistance, so looking for convergent resistance mechanisms between drugs, is valuable, again, for kind of biomarker and novel drug target reasons. So what would we want a method that enables you to measure resistance to look like? So here's a kind of this is again a kind of highly simplified schematic of what we would expect a population to do over time when we repeatedly expose it to a given drug. You know, we might expect that if the nature of the drug means you have to give it in these cycles, that you would initially kind of kill fractions of cells, but over time you would see resistance emerge, and that would be realized as an increase in the population size. So ideally, for our method, which enables us to measure resistance evolution, we would be able to just look at the sensitive and resistant cells in that population, and we would track this resistant population as it emerged, and we would fit the kind of observations of that population to models we've developed, and we could quantify key behaviors about that resistant subpopulation. Now, of course, the problem is that in the first place, we often don't know what the mechanisms of resistance are. And so there's no easy way for us ahead of time to track what that population is doing. And that's just another way of saying there's no easy marker of resistance in the population. So if we can't do this, what can we do? And for this, we turn to these lineage tracing techniques. So instead of now trying to track specific phenotypes that we don't know ahead of time, what we can do instead is track the fate of individual cell lineages. And just briefly, these techniques work by introducing a stable neutral tag into populations of cells, and this is if you're using them experimentally. And by taking samples of those populations over time, you can sequence these tags, and they give you a readout of the cell lineage composition and how it's changing. And you can think of this as essentially as a readout of the evolutionary dynamics over time. So now you can see in this hypothetical scenario that whilst we don't have a readout of the specific resistance mechanism that we're interested in, what we do have a readout of is this rare subclone, which has been, in this example, labeled with the purple barcode, slowly expanding as we repeatedly treat the population with a given drug. And what I'm trying to emphasize here is there's actually information encoded in the distribution of lineages that survive, which tell you something about the underlying dynamics. So whilst you might not have a specific marker for a resistance phenotype ahead of time, what you can do is look at how these dynamics differ and start trying to understand resistance from a population down, from the top down. Okay, just sorry, checking how we're doing for time. So this is a kind of really obvious example, and this is what the kind of typical model of resistance evolution is this kind of stringent bottleneck followed by a clonal expansion. But there's been lots of work recently showing that actually resistance can often emerge via non-genetic switching states. And so in those cases, you might expect those lineage distributions to look something more like this. So hopefully you can see that the total population is expanding at a similar rate, but if we look inside the cell lineages that are evolving, we can see that there's now a high diversity because cells have a high propensity to switch between sensitive and resistant states. And so the resistant phenotype is now distributed amongst many cell lineages. So hopefully what I've tried to emphasize there is that there's information captured in the distribution of the cell lineages that survive that can tell us something about the underlying dynamics. And so this is what we use in the lab, or we've developed a method to essentially leverage that information to quantify the underlying dynamics. And that's hopefully moving us some way towards having a method that allows us to quantify resistance evolution in a preclinical setting. So again, I'm sorry that there's a version of this figure which has a lot more detail on where we would go into the kind of more technical aspects of how the method works. But in the interest of time, and so that we've got time for kind of questions afterwards, I won't go into the details today. But I'd say these are the kind of key components of the method, and the method is we've called evolutionary informed resistance assays, or IRAS, and this is kind of the logo representing it here. So the first step is we've developed these computational models that capture key aspects of how we expect resistance to evolve in the population. And if you're kind of familiar with the mathematical modeling of resistance field, they have lots of features that you would recognize. So for example, we model the pre-existing fraction of cells that can be resistant, so that some parameter would encode that behavior, and we encode the differential growth rates of the two phenotypes, so sensitive and resistant cells. We permit transitions between them, and all of those different values, capture some hypotheses about how you expect resistance to evolve in the population, and we then take the outputs from our experimental evolution experiments, which are those barcode distributions. So here I've shown an example data set from this lovely data set we've got from breast cancer, where we've looked at some CDK4/6 inhibitors. And we take those distributions and we fit our models to them to infer what those key parameters are. And what this enables us to do is get a quantitative readout of what we summarize normally when we talk about as a propensity for resistance, but you could break it down as these different behaviors. So how likely was it for a cell to be resistant at the beginning of your experiment, and how easy was it for cells to transition between sensitive and resistant states? And so this is what this kind of output of the method here is trying to highlight. We can somehow quantify resistance evolution, and hopefully you can see there's a reference to the Nature Comms paper there if you're more interested in some of the technical details about how this method works. So, I've described this method we've developed, which enables us to quantify resistance preclinically. And zooming out again, how do we envisage this fitting into the drug discovery process? And I think we're kind of breaking down the outputs into these three key things. So I think the first one is the ability to actually quantify the propensity for resistance is important, because it enables you to rank different drugs by their propensity for resistance. So you can imagine in a hypothetical drug development scenario where you're trying to decide which compound you might take forwards, if you had some readout which could tell you there's something inherent about the biology of that compound, which means it's more or less likely for resistance to develop, why that would be valuable, incorporating that information into the drug discovery process. We also think it could accelerate the identification of the mechanisms themselves. So this ability to distinguish between these cases where resistance is the product of a stable genetic mutation versus these transient non-genetic switching mechanisms can actually help guide what functional assays you do to profile the resistance mechanism, And then finally, this kind of slightly newer idea we've been looking at, which is looking between drugs and quantifying the kind of levels of cross-resistance between drugs, which is a kind of new functionality we've been working on of the method. And being able to rank drugs by their cross-resistance strength will be able to enable you to hone in on these strong drivers of cross-resistance between drugs. And we think that has repercussions for drug discovery and for discovering new biomarkers as well. So
Brilliant.
That's great. I think that's the end of my slides. So I'll now pass on to Michelle, who will tell you a bit more about some kind of clinical applications of these.
So that's
evolutionary
application
ideas.
right at the beginning and now right at the end.
Okay, thanks. Yeah, thank you for the introduction. So I'm going to switch gear and talk a bit more about how some of these concepts are being evaluated in the clinic. So the first thing to point out is that as clinicians, we generally dose anti-cancer drugs according to what's called maximum tolerated dose, so the highest dose that a person can stand. And drug development pipelines work towards establishing what this dose is. However, as you've heard, this strategy usually fails in metastatic solid cancers, and perhaps that's because of pre-existing drug-resistant subclones, as Trevor highlighted, or perhaps it is the kind of plasticity that that Freddie's just been talking about. But either way, there's a real paucity of available anti-cancer drugs, which means that we get stuck quite quickly in the clinic. So just to illustrate this with one of my patients, and here you're looking at her tumour marker, the CA125, plotted over time as she proceeds through therapy. The dotted line indicates sequential lines of chemotherapy treatment. So you can see that when she first comes to us, she responds quite well to chemotherapy. Her CA125 goes down into the normal disease, and into the normal range. And so she has clinically undetectable disease. But then after a median of about 18 months, her CA125 starts to increase again. She's relapsed. We treat her again, and the rest of her disease course is characterized by these this intermittent treatment schedules. But each time we give her chemotherapy, it works a bit less well. The nadir is less deep, and the time until she requires treatment again becomes progressively shorter, until ultimately she dies with effectively untreatable cancer that is dominated by resistant clones, and that happens at a median of 4 to 5 years. And the other thing to point out here is that each time we've treated her, we've used the same drug indicated by a P for platinum. So in a sense, it's no great surprise that she ultimately becomes resistant. So why does this happen? Well, new drug development is slow. So when we look at the agents that we've had available to us for treatment in first line platinum sensitive cancer, this is all we've got over several decades. And when we think of the amount of effort we put into recruiting women and all of the women who, you know, contribute their time to clinical trials at the end of their life, it's slightly disheartening. We've tried increasing chemotherapy doses, using more complicated combinations, treating relapse disease earlier, immune checkpoint inhibitors we've tried in our disease, but sadly none of these have been effective. And you can see these studies are old, so we haven't really improved on this situation for first-line treatment of ovarian cancer for a number of years. So I became interested in these kind of evolution-based approaches as a potential solution. And I'm going to talk to you today about one therapeutic approach known as adaptive therapy. So this is sort of based around a premise that, that evolution is subject to trade-offs. So this means that resistant phenotypes incur some kind of a cost. So for cancer, we might say that resistant clones proliferate less well. And so it follows from that the relative fitness of sensitive and resistant clones is sort of flipped by therapy. And so this is a similar diagram to the one that Trevor showed earlier. So you can think on the top arm of this cartoon, if you treat patients with repeated rounds of maximum tolerated dose chemotherapy, you will progressively eliminate the sensitive cells. And that means that by default, the resistant cells now become more fit. However, what adaptive therapy aims to do is to prescribe treatment dose interruptions or dose modulation with the aim of maintaining some population of sensitive cells that can outcompete the resistant clones. And it's important to point out here that maximum tolerated dose does aim to cure and normally fails, whereas adaptive therapy acknowledges that we can't very often cure cancers in many situations. And so the idea here is to achieve some kind of stability and control patients' disease over the long term. So I think for our patients, if we can extend the x-axis on the previous graph I showed you, that's actually quite a big win. And people have begun to test this. This is about the most sort of mature example, if you like, led by Robert Gatenby and the team at the Moffitt. So this is a trial of men with castration-resistant prostate cancer treated with the oral therapy abiraterone. And you can see on the trial cohort in the top figure that when they have a reduction in their tumour marker, then they have a period off the drug. And so you can see these men have frequent dose omissions. They were compared to a cohort of contemporaneously treated men who received continuous abiraterone, and you can see that in all cases, those men having standard therapy progress quite quickly. So in the experimental arm, progression-free survival was extended and the men had less drug overall. A criticism of that study is that it wasn't randomized, but the randomized trial is happening in the US and in Europe, and that has now finished recruitment. That's the ANZADAPT trial at the bottom. There's another example of an adaptive therapy trial in melanoma that is currently underway. So we were interested to see if we could apply this in ovarian cancer. This is a figure from a paper. You can see the reference at the bottom that can give you more detail about what we did. So it's a slightly complicated figure, but these are all mice with ovarian cancer tumours. The top row in green, those mice have the more sensitive tumours. The bottom row in red, they have the more resistant tumours. And then the blue ones in the middle are a mixture of sensitive and resistant cells. And then in the columns on the left, you have the vehicle-treated animals, and you can see they all progress and die. And then the middle column shows the standard therapy, and that's indicated by the grey bar. So if you look at the most sensitive green cells at the green graphs at the top, you can see that the treatment sort of stabilizes tumor growth. It's not a terribly sensitive model, so the tumors don't really shrink. But then as the treatment is withdrawn, all of those tumors rebound and grow back. And that happens as you go down the column more quickly as you introduce more resistant cells into the tumors. However, the adaptive therapy arm, in which we modulated dose based on changes in the tumor burden, was able to achieve long-term tumor control as we had sort of hoped from the schematic I showed you in the last picture. And I just want to point out the adaptive therapy regimen. So you can see that when the tumor volume increases or decreased by more than 20%, we make quite a marked change in the dose, increasing it or decreasing it by 50%. And just to emphasize it, it improved the outcome for those mice. An observation we made in this experiment was that adaptive therapy did increase the cumulative drug dose overall. So on the graph on the left, the dotted line indicates the amount of treatment that the standard therapy mice received. And each line on this graph shows a different adaptive therapy treated mice. And you can see that the cumulative dose is higher for all of them. The other thing you can see is that the graphs for the cumulative dose plateaus over time. And what we found was that we were giving the mice repeated very small doses towards the end of the experiment. However, because they live longer with adaptive therapy, the dose per day was not greater with adaptive therapy, and the mice tolerated it well. And this is just showing that their weights were stable over time. So based on these data, amongst other things, we decided that we would like to test this in the clinical trial. So we thought, because we have a tumor marker and because we use platinum regularly, that it was a reasonable thing to do. So we wanted to test in women with relapse disease, because this is a very experimental approach. They had to have some degree of platinum sensitivity, and clinically we defined that as no platinum for the previous 6 months. We thought it had to be reasonable not to give combination chemotherapy, which is often preferred, with some quite weak evidence in favour of that. We wanted the trial to be randomised, and because this is so new, we wanted to include some translational endpoints. So I just want to quickly show you the adaptive therapy regimen that we settled on. So you'll recall from the mice, it was a 20% change in tumour burden for a 50% dose change. Here we settled on a 25%, so a bigger change in CA125. And this time we're reducing the dose essentially by changing the dose by 20% each time. And this was based on some data that we had of CA125 taken from women treated with single agent carboplatins, courtesy of Charlie Gawley, who may be on the call. And so we based it on that. And then we had to really work hard with our investigators who were recruiting patients around the country, our patient representatives, the ovarian cancer charities, to settle on a regimen that was acceptable to all of them. And so this may not be the perfect adaptive therapy regimen, but it was the best we could come up with. And yeah, just illustrates the constraint that we have in clinical trials. So we're powered to detect progression-free survival. We have a large number of secondary clinical endpoints, including cumulative carboplatin dose based on what we learned on the mice. We are open in 13 hospitals around the UK with another 2 in setup. We've got 34 patients enrolled so far. And our translational program, we've costed to collect 1,500 samples. These are sequential samples from our 80 patients, and we have 500 already collected. So the translational work to try and understand this mechanistically is coming. If we have time, I was quickly going to present a case. I'll show you one of the cases. So this is again courtesy of Charlie Gawley. So it's a bit of a statistical quirk that none of the patients I've recruited have been randomized to adaptive therapy. So this is from Charlie. So this is a woman presented back in 2014, had the usual, high stage disease that we normally see and the standard surgery that our patients would receive. So she relapsed in 2020, then had combination chemotherapy, and then relapsed again 3 years later, enrolled into Actov. So I just really wanted to show you what happens to her carboplatin dose. So she starts at the highest dose. The AUC is just how we calculate dose based on renal excretion. So she started off with a high CA125 of 690, received AUC5, and then you can see with sequential cycles that AUC, the dose of the carboplatin, goes down by 20% each time, until in the end she reaches the point in which we actually omit a dose, and you can see her CA125 responded. So this, I think, is similar to what we saw in the mice. sadly, she had a complication of her cancer and had to come off the trial. But yeah, I think it is sort of, for us as clinicians, this is really surprising. And we expect that we should be giving high dose continually, and that when we give small doses like this, it's not effective. So yeah, an anecdote at this stage will be interesting to see what the trial shows up.
It's remarkable. Yeah. Thank you.
That's the end of me.
Yeah, I think it's really exciting where this is going. I can't wait to see the trial results. So
Yeah,
thank
really
you very
nice.
much. Yeah.
Thanks so much for that that tour from, conceptual to preclinical, and then with the clinical trial, and very much we'll be following that and staying tuned for that. Um, I'm gonna cycle through chat questions as best I can here, and I'm sorry if I missed anything. So put it back in if I've glossed over it. But I'll start with Dr. Maley's question. I think this is for Dr. Whiting. Does that barcode plot come from a constant dosing or periodic? So just clarification on that.
Yes. So, hi, Carlo. The example I gave, which was the real data, came from continuous treatment. So that's on CDK4/6 inhibitor treatment. So then it's safe to kind of leave the cells on continuous treatment. We found that we tend to prefer the dosing regimens where you don't have to slowly ramp up the concentrations over time, just because I think, especially since we're interested in the evolutionary dynamics, that might lead to qualitatively different dynamics than having acute exposure either continuously or periodically, which is presumably more like what patient tumors would see. So we try and focus on those cases. So yeah, that was continuous treatment with a high dose of CDK4/6 inhibitor in vitro.
Thank you. And then a couple of questions I think that lump together about, the non-genetic contribution to resistance. And I don't know if anyone cares to, I'll keep it that vague, but does anyone care to sort of take a stab at what might be the non-genetic contribution and how we might get at that? Like parsing what's genetic, um, a genetic driver event versus like non-genetic plasticity, and how we can study those dynamics.
Yeah, should we go along? Do you want to start?
Yeah, perhaps I'll go first. So I think a great example is to think about chemotherapy. So chemotherapy has been given to many types of tumors for many decades now. And yet still, I think in virtually all cases, we don't know what the resistance mechanisms are to chemotherapy. And that's because typically we've done sequencing studies where we sequence pre and post therapy with whole genome sequencing, say, and look for differences. And we just don't find, and many people just haven't found consistent genetic alterations that you'd expect to see if there was genetic resistance there. So we strongly suspect it's therefore non-genetic. Of course, it must be heritable. Otherwise, the tumor would instantly lose its resistance again if it wasn't heritable. So we think, therefore, it's likely to be in the epigenome because that is quite heritable.
Yeah, just to extend from that, I think again, some of the barcode data we see, there's these are cells that have been exposed to an acute concentration of the drug for up to a year, and with replicate flasks that have been separated, over 300 days ago, and yet you see the emergence of the same stable subclones. And then yet when you do the functional work to look for the resistance mechanisms, this is again in some of the CDK4/6 work, there's no smoking gun, there's no genetic driver which points towards what that mechanism might be. So I'm interested in those cases because, as Trevor says, it points to something heritable, which obviously I think if you're talking about putative drug targets, it kind of by definition needs to be heritable. You don't want to target something that's really transient. And so I think that those data are exciting because they point towards these kind of incredibly stable and yet non-genetically mediated mechanisms. So yeah, I'm not sure what they are in a lot of those cases, but I'm kind of excited to try and work out what they are.
Yeah, thanks. I would add to that. I mean, just to chime in my own sort of thoughts on this, like the pressure imposed by the change in ecosystem, I mean, essentially by drug, I think can make a non-genetic response seem heritable in the sense that continuous pressure creates an environment necessary for a non-genetic mechanism to remain on. And so I think, as long as there's drug present, that population is subject to that constraint. And plasticity can play a role there. But anyway, I think,
Yeah, I
just
think that's right. I mean, maybe Michelle, you want to comment, but I just everything we said is so tumor cell centric. But of course, these drugs are systemic, so they affect all of the cells in the body. And
right,
Yeah, I mean, so we're very interested in that and this kind of idea about how an environment can be supportive of a resistant clone. And I think work coming out from our lab is implying that, again, is quite intuitive, that probably the resistant clones are able to sort of create a favorable environment for them to live in.
right,
It's very early stages, but we definitely see that a resistant matrix can protect cells from chemotherapy.
right. Yeah, I was gonna ask Bernie if he wants to hop on and ask his question. Then Carlo had another one. Maybe you can pop on after Bernie, if you're willing to. Otherwise, I'm happy to answer it or ask it on your behalf, Bernie. Oh, you're muted, sir.
Right, sure. Thank you for that. Um, it seems that the key to the adaptive therapy is the trade-off. So I'm wondering, what, if anything, do we know about the trade-offs between growth and resistance? And it seems like, to the extent that we know something, then an independent approach would be to find ways to sharpen and increase the magnitude of the trade-off. So can we speak to that at this point?
Yeah, so I mean, in the lab, we've shown that there is a trade-off using, I mean, it's using established cell lines in which we've evolved resistance in different settings, either in vitro or in vivo. So we do see the trade-offs certainly in that preclinical setting. In terms of whether they're there in people, of course, that's much more challenging. And it's one of the things that we hope to get at through the ACTor translational program. So our women are providing I mean, it's back to the sort of the genetics of cancer cells, but they're providing blood for us in Streck tubes with every treatment cycle and then very frequently in follow-up. So we will have a large longitudinal data set from that. And also some repeated biopsies, which is of interest to me to understand what is that, because, to understand what's happening within the tumor microenvironment. In terms of how to target, I suppose you're asking how to increase the trade-off therapeutically. Sounds challenging. I'm not quite sure I have any ideas for that yet.
there's one idea that I'm sure you're familiar with, Bernard, that I'm quite obsessed about at the moment, which is this idea of using hyperactivation of oncogenic signaling as a counterintuitive way to treat tumors. So we're well aware, of course, like lots of targeted therapies shut down signaling, and we know that that's toxic to cells. But it's also lots of evidence that if there's too much signaling in a pathway, that's also toxic to cells. And so there's ongoing drug discovery programs about essentially drugging, inhibiting the negative regulators of a pathway to increase the signaling as a way to kill cells. But great in its own right, but I think it's interesting in the context of resistance as well. So if we have resistance to an inhibitor, to be resistant, that cell needs to turn on oncogenic signaling. And so therefore, if we could then further increase the signaling, you'd imagine that the resistant cells, which already have elevated signaling, would be particularly sensitive to those activators. And so you could imagine having cyclical activator versus inhibitor treatment as a way to exactly potentiate that trade-off that you're suggesting.
I think,
Right,
yeah.
thank you.
I think
That's
I would
very
just add
cool.
that, yeah, I think you can conceptually think of the cost in two ways. There's the kind of, I think, more classic metabolic cost, as in there's no, the trade-off idea is in the metabolic capacity of a cell is limited. And so if you're investing in efflux pumps, for example, you then have limited kind of energy to spend on other things. But then the other one is the signaling way of framing it, which I think a lot of the original observations to do with the cost were this oncogenic addiction idea, which is that the resistant state has become reliant on the cost. I also just add really quickly that I'm sure other people on the call will know better than me, but I think there has been some theoretical work that shows in some scenarios adaptive therapy works even in the absence of a cost. So, for example, I think if there's kind of phenotypic switching, it can work, and also there's phenomena to do with the kind of carrying capacity. But yeah, I'm sure other people on the call will know much more about that than I do.
Yeah, I think I wanted to highlight the efflux pumps thing because as you're saying that, Freddie, Bob is talking about the efflux pumps. And so not that Dr. Gatenby needs more credit for good ideas, but he should get them when here too, among the many that he's gotten. But he suggested to us, we were looking at efflux pumps as a mechanism of resistance. He suggested this exact thing that Trevor and you were talking about with hyperactivity of a pump. So give substrates to the pumps that ramp up their activity, increase or exacerbate the fitness cost of that pump being active. So I think I'm glad he put that in there. But
Yeah,
Carlo,
it's a great idea.
yeah, it's a fantastic idea. Well, you said it as well, so really interesting. Carlo, did you want to hop on and ask your question about intermittent dosing and
Sure, and I could just do a follow-up. As Jason probably knows, we actually have submitted a paper to the special issue of a review about what is the evidence for a cost of resistance. And I was surprised by some of the results. Unfortunately, it's not near 100% in terms of the competition experiments that have been done. And the competition experiments you saw today were in that review, the one from this group, Trevor and Michelle et al. Um, but so I think to answer Bernie's question, I don't think we even know in general how often there's a cost of resistance, let alone what the specific trade-off is, except in a few cases like Bob's work and the efflux pumps, which were using up, I think, half of all the ATP in the cells in that one particular cell, MCF7 cell line that was resistant to, I think it was docetaxel or something. Anyway, my question is a little bit different. It's about if you do the intermittent dosing and you're fitting your math models to both the regime without drug and with drug, then I'm guessing you can estimate the cost of resistance and how the two populations behave in those two different regimes. Do you see that and have you measured the cost of resistance in more than one cell line or drug?
So I've seen a few data sets where there is compelling evidence for a cost at the end, but that's usually measured in a kind of the simpler way, which is essentially just doing some IncuSite experiment where you measure the growth rate of the evolved resistant cells. We found that trying to fit it directly fitting the models directly to the lineage tracing data is that if you have a model that permits both a cost of resistance and some sort of phenotypic switching rate where you can switch back from resistant to sensitive, those leave often kind of indistinguishable signatures in the lineage distributions, which is just that in the cost case, they haven't grown sufficiently to have expanded to as larger lineages that you would expect. And obviously in the switching case, the ones that have reverted to sensitive are killed by the subsequent drug. And so the output's the same, which is a smaller lineage size than you would expect. And so we had kind of problem, and I guess there's a link there to the kind of theoretical work showing that these cases where you don't need a cost, so for example under the high reversion switching rate, that's probably why that's true. So yeah, the short version is no, we've struggled to directly quantify the cost just using the barcode data.
Going forward, one of the things we're doing is, and I think others are looking at as well, is to collect very large sets of patient data where there's longitudinal monitoring of tumor burden, and there's intermittent therapy just for, not necessarily in an adaptive trial like Michelle's trial, but just because of other clinical and personal factors. And then so there's lots of periods monitored on drug and off drug. And we hope with enough data, with enough heterogeneity in the treatment scheduling, we might be able to learn some of these costs directly in people as well.
I think Bob's asked you a question that chat, Michelle. Do you want me to, he says, Michelle, do you want to mention the change in culture that you used to show the cost of resistance?
Yeah, sure. So an important kind of concept is that costs are only going to be revealed when resources are constrained. So that's kind of central. And so we played around, and others have done this as well, played around in the lab with constraining things in the culture medium. So of course, in general, when we're culturing cells in the lab, we give them an abundance of everything they need, space and nutrition, nutrients, etc. So we have tried reducing serum, which is difficult to sort of link that to what happens in patients. We've also tried reducing glucose, limiting space, and all of them expose a cost of resistance in different settings. When we don't do that, we don't see a cost of resistance at all. And then when we see, when we look in mice, we see a cost of resistance, presumably because within the tumor microenvironment, we know that there is a diminished blood supply. Is Bob asking me some more things?
I think he's agreeing and
Commenting,
warning people.
commenting about oxygen, oxygenation and amounts of glucose in standard culture media.
Yeah.
So, yeah.
Yeah, sure. No, absolutely. So we have reduced glucose in the culture medium. We haven't tested hypoxia yet.
Yeah.
Interestingly, sorry, I'm losing the ability to speak. A lot of the hyperactivation results show similar trends. So a lot of these being able to look for effects of hyperactivation of oncogenic signaling leading to kind of apoptosis and those sorts of things only emerge if you have a stressed cell culture condition. So there's not a bunch of growth factors which mean the cells are happy. So just that's interesting as well.
Yeah. Michelle was going to ask maybe maybe it's borderline philosophical, but I think there's like very clear preclinical evidence and some nice clinical evidence for at least adaptive therapy and some steering, sequential dosing concepts. Why do you think, like, if you had to point to a barrier to adapting these types of paradigms in a more widespread way clinically, what do you see in the clinic as being some of the barriers that you encounter? I mean, are they more on the physician side? Are they more on the patient appetite side? I mean, what sort of things do you encounter?
So if I talk about patients first, so in my practice, I haven't found patient barriers to adaptive therapy. There are sometimes patient barriers to participation in a trial, but in general, they like the concept. I mean, in my patient population, as you saw from that graph I showed, we know that we can't cure them. We tell them that a bit at the beginning. They receive frequent kind of repeated lines of chemotherapy, and they know that it's not going to cure. They're used to that kind of sequencing, if you like. So I have found that patients in general quite like the idea, a possibility that it might extend their survival and less drug, less toxicity. So I haven't identified patient barriers. I think we have to bear in mind that this is really in its infancy. Bob's trial that I showed is the first clinical trial. I think our trials are just coming, and the numbers of trials testing this concept remains small. So until we start to see some benefit in randomized clinical trials, I think that will be the thing that switches the field, to be honest. I mean, I can be philosophical and talk about what it's like to what it might mean for drug companies. They may have a different opinion where they've got their motivation may be slightly different, but yeah, I think we just need to wait for the clinical data and see if we see benefit and understand a bit better how this is working and what are the situations in which to use these strategies.
Do you think there's another barrier around, I guess, perhaps with drug companies, that often we're talking about using drugs that have been around for a long time already, whereas a lot of the trials are geared up to testing new agents in new settings?
100%. And funding those trials is really challenging. We're looking at academic funding and academic led trials, which is always challenging compared to pharma studies. So yeah.
Thank you. Lots of discussion going on. Dr. Sendek, would you like to chime in and ask your question? Please feel free. Okay. I can say it. Do you think cell-to-cell communication between cancer cells helps some or few of the cells to prepare for the drug? Doing something maybe phenotypically to them. Certainly pumps, right? I mean,
I mean,
transfer mitochondria is one that people are looking at, thinking about, at least for metastatic niches. So I would imagine similar. I haven't seen with drug in particular, but I know there's mitochondrial transfer that may be happening.
We've sort of looked at it a little bit. So we've tried in some of our in vitro systems to look to see if they're particularly sort of secreted factors, and we didn't find any evidence for that. Just an observation we made that is of interest to me is that when we that when we had co-cultured tumors in mice, so injected with different, mixtures of sensitive and resistant cells at different ratios, we were quite surprised to find that over time the resistant cells kind of grew as little islands embedded within otherwise sensitive tumors, which we haven't yet explained that, but it's of interest. Why do they form these kind of little nodules within a tumor? Why aren't they spread equally within the tumor? And I suppose a question that we have is back to my previous point, that they are sort of constructing a niche where they can live happily, but whether they're communicating between each other within that niche, we don't know yet, but we certainly see them localizing together.
Yeah, well, I would just say to add to that, we see the same thing. I mean, we'll label sensitive and resistant prostate cancer cells completely separately, put them in a co-culture, a 3D spheroid co-culture, and the whole inner spheroid will be resistant, the whole outer will be sensitive, or the whole I can't remember which direction it is, but it's clear separation. And I've always thought it's probably, which other people have shown, it's probably just the cadherins that it's easier to bind to each other than to the other culture, the sensitive cells. So that's been kind of demonstrated. That's always been my thought about it. But we see the same exact behavior. They just like each other more than they like the other partner. So yeah, really wild. Yeah. Yeah, I was curious, Freddie, if you've done release experiments. We've been really so both release and have you looked at the population or the response to drug very early? Because what we see, our observation, and I'm just curious if you see similar things, is the first pulse of drug, the biology of that response is completely different than the biology of the adapted response 6 cycles later, 5 cycles later. The first pulse, that response is very different in its biology. If we then release those pulses of drug for a month to 6 months, the resistance mechanism whatever it is, remains intact, but now they gain proliferative advantages over early resistant cells. So they not only do they maintain the resistance, so they have some memory, quote unquote memory for this, but they now have this added benefit of they're more proliferative than their early resistant counterparts. So I was just curious if you kind of looked at the dynamics of that behavior over both early response to drug resistance and then release from it.
Yeah, so we have looked at that either. So in my work, we tried to look at that explicitly because we were conscious that, especially with chemotherapy, recent exposure to the drug would leave like a transcriptomic footprint, which presumably is something to do with how stressed the cells are from having been recently exposed. And we wanted to be able to tease apart the transcriptomic profile of actual resistance from that. So we had another condition, which was they'd been exposed to like one cycle of the drug. And then we profiled those, and that was presumably the majority sensitive cell gene expression signature of chemotherapy stress. And then we had the resistant cells at the end, and their gene expression profile would have been presumably some composite of that stress plus whatever the resistance gained. And we tried to do some differential expression between those to isolate them. I say that when you have the barcode data, you can normally distinguish quite a few of those things. So we found that normally what you're looking at on those early pulses is the signature of the majority sensitive cells dying off. And so if you look at mechanisms at that stage, that's what you tend to be looking at. And then in these cases where you get a really strong clonal bottleneck, you'll see the dominant barcode at the end will have gained some capacity, which means despite that kind of stress to the drug, it can continue to proliferate. And that's what the resistance looks like at the gene level. So that's how we've tended to look at that. We've also found cases where you still get a strong bottleneck, but the bottleneck's probably due to just the stochastic sampling of the few cells that happen to survive, because when they grow back, they're as sensitive to the drug as the initial population. So that's probably more reminiscent of these persister dynamics where the survival over time is to do with your ability to survive a single cycle, not multiple cycles. So hopefully that answered your question some way.
No, that's great. Really interesting. Yeah, thanks. yeah, I think we see both depending on what the drug is, what its mechanism is. We see persister behavior in things that are cytostatic, like enzalutamide. We see um, like a metabolic or an energetic, not metabolic, but an energetic difference in things like that are, cytotoxic chemotherapy. Um, that's been sort of my favorite answer to this whole thing lately is that the both the plasticity and the memory for quote unquote memory for resistance and drug is got to do with the energetic differentials, and how drug sort of steers that resource acquisition and energetic demand, that includes pumps, cell division, etc.
Yeah, we find that chemotherapy just tends to be messier. So I quite like the idea of having a mechanism at the end that you could target with a drug, for example, and that tends to
Agreed.
not be the case in chemotherapy. But for targeted therapies, you
Yeah.
tend to get a nice clean mechanism, which is nice.
Yeah. Well, thanks for the great discussion. Thanks everybody for your attending. Thank you to the speakers. If anyone has additional questions, comments, we have a moment, but otherwise, find them offline, I would imagine. I would imagine they're responsive. They seem to be in planning this.
We are. Yeah.
Thanks,
Thanks.
everyone. Thank you. Really nice.
Thank you. Great talk.
Thanks.
Good to see everybody.